A disease can prevent you from being able to sleep was known for centuries. It was named fetal familial insomnia in 1986. It derived from a family in Vinodol, Italy. The book, "The Family Cannot Sleep", described very detailed. It has 4 stages. It can cause psychiatric symptoms such as panic attack, paranoia, hyperactivity, and mood symptoms. It also can cause increasing of cortisol level, heart rate, blood pressure, body temperature, breathing, and perspiration. The patient is completely disruption of sleep, inability to speak, and dementia. There is no treatment or cure, so it is very terrible for those patients. It is very rare. Fetal familial insomnia is caused by a type of protein called prion protein. All mammals have a prion protein. It is encoded by PRNP gene, which is on short arm of chromosome number 20. The proper form of this protein is C-prion, and misfounded CS-prion is toxic for neurons. Once introduced into body, the CS-prion binds to C-prion and converts it into CS-prion. This leads to CS-prion aggregates into the brain. There is a difference between normal prion and causing disease prion. The former is highly alpha helical, whereas the latter has a larger beta sheet. That indicates the normal one is more polar. There are 2 important ranges in this picture. One is between the beta strand 2 and the alpha helix 2. It is considered to be potentially involving C-prion to CS-prion conversion. Another one is here. It is relatively unstable. When the disulfide bond here is reduced, these 2 hydrogen bonds will be broken, and the alpha helix tree is able to swing to the opposite direction. There are 2 human prions here. One is on the left, one is on the right. They swap their alpha helix tree. The crystal structure of human prion protein is dimer. As previous slide described, the intra-chain disulfide bond must be reduced. 2 alpha helix tree must swing out across the dimer interface to swap and back again, and the 2 disulfide bridge reform. The swapping is a geometric conformational transition. It is a mechanism of oligomerization for a variety of proteins. However, some peers suggest another form of dimer. The 2 antiparallel beta sheets can form a 4-stranded beta sheet. In 2020, scientists discovered that a single-point mutation goes to valine at 127 in human prion may prevent prion disease, but structural basis for its protective effect remains unknown. They show that the mutation alerts and constrains the prion backbone conformation by increasing intermolecule hydrogen bonding. It also changed the solution geomatics of the beta 2 to alpha 2 loop. So the hydrogen bond in here is more tighter than here. It changed to more soluble. It seems two different dimers and two different mechanisms to me. One tends to decrease polar, have the potential to mix CSP and cause disease. The other one does not. Fatal familial insomnia is neurodegenerative disorder, Alzheimer's, Parkinson, spinal muscular atrophy, Huntington's disease, BSE, CDJ, and CRU are other neuron-like disorders which have a similar mechanism.